Photosynthesis is magic. I've said it before and I'll say it again, but it's not going to earn you points on the AP Biology exam. Hi, I'm Miss Parrott, and in this video, we're going to be getting into what will earn you points on the AP Biology exam. All of the nittiest, of the grittiest, deepest dive into photosynthesis. So we're going to be following the electrons in photosynthesis. This video is paired with following the electrons in cellular respiration and my other chemistry fundamentals videos. The puzzle we're going to be solving today is how do we turn light, which is not matter, it is energy, into actual matter. What? This puzzle totally changed the direction of my life. I'm not even joking. I used to be a theater major, and learning about this totally, like, I became a science major and now I'm a science teacher. So let this blow your mind. All right, if we are following the electrons in photosynthesis, first let's flash back. What is our basic equation? Water plus carbon dioxide yields glucose and oxygen, right? So where do the electrons, since we're following the electrons, where do they start? Well, we get our electrons from water. Water, that first reactant, if we took 2 water molecules, that's 4 hydrogen atoms and 2 oxygen atoms, we can yield, through the process of photolysis, 4 electrons, 4 protons, and 1 O2 oxygen gas molecule. And all these are going to go different places and have different jobs. So water is our initial electron donor. It gets oxidized, right? Oxidation is losing electrons. And P6AD+, which we're going to get to in a second, is the physical molecule that gets reduced. Reduction is gaining, that gains these electrons. Are you ready to go down the rabbit hole of photosynthesis? Let's do it. Whoa! I'm not stopping. Okay. So, the very first thing, it's like, what is light doing anyway? Light's job in photosynthesis, it's the photo in photosynthesis, is to raise the potential energy of electrons, the electrons that came from water, to a higher energy state, higher potential energy. So how does it do it? Let's look at the machinery, the structure of the thylakoid membrane. So in our picture, up here is the stroma. That's like so we're in the chloroplast. It's the outer part of the chloroplast. Then you have the thylakoid membranes, right, which are like little pancakes. And so this purple is a thylakoid membrane with these blue embedded proteins. These green are chlorophylls, okay? And as we know, chlorophyll is really important in capturing light. Oh yeah, that's right. Photosynthesis. Yeah, that's what we're doing. Then inside the thylakoid membrane is the thylakoid lumen, also called the thylakoid space. I will be using both interchangeably in this video. I'll usually say lumen more, but your book may say space. The AP Bio is going to use lumen, so keep that. Those are analogous, synonymous, if you will. All right, so first, let's look at the structure of Photosystem II, because Photosystem II is a protein complex. Like when you learned about quaternary structures of proteins, this is a quaternary structure. It has lots of different parts. So on the outside here, you have embedded a bunch of chlorophyll molecules. Chlorophylls are, and they're even called like chlorophyll antenna pigments, because they are what are receiving the photons of light from the sun. Then you also have in the middle of the photosystem II is called the reaction center. In the reaction center is a chlorophyll dimer, or like a 2-pack of chlorophyll. You can think of it, 2 chlorophylls stuck together. And that's called P680. P680 can be reduced as P680. If it's oxidized, in its oxidized form, it's P680 plus. And so it is going to, so water is going to, right, oxidize. It's going to give its electrons to P680 plus. And when it does that, it's going to get an electron and just be P680, because the negative electron will neutralize the plus. Hooray. So now P680 has electrons. Farther on in that comes the electron transport chain. But let's just focus on photosystem II. So this raising the potential energy happens in a really cool way. Now, I think of it, this is my tennis racket, tennis player analogy. So imagine you are a tennis player. You have a racket in one hand and a tennis ball in the other hand. And you're just standing there like this. Okay, so let's imagine that the sun, instead of shooting photons or packets of light, it's shooting tennis balls. Okay, so imagine it's shooting tennis balls, and one of those tennis balls hits your tennis racket. Now, your tennis racket is actually going to be, that's analogous to one of these antenna chlorophylls. Okay, now it is going the speed of light. So even though photons, I could do a whole video on light and photons, but even though it doesn't have mass, it has energy and it has momentum. So when it hits these antenna chlorophyll, it physically causes them to vibrate. They physically start moving. Now imagine if something hits a tennis ball hits your tennis racket so hard, you don't just feel it in the racket, your hand feels it, then you feel that up your arm. Now imagine it hits so hard that it's like it sends all the way to your other arm so much, pop, that tennis ball you had in this hand pops out. That is exactly how photosystem II works with light to raise the energy of electrons in P680 to an excited or high potential energy state. This like that where light is passing that energy from one chlorophyll to another is called Forster resonance energy transfer, or FRET. So that's really cool. So electrons are not being passed from one chlorophyll to another. It's the vibration that, like imagine you could get your whole class together, hold hands and do that like worm thing. It's like that, right? Like one person pulls your arm up and that's gonna make your arm go up and it's gonna flow through the whole squad. Teachers, that's a very good, fun activity to do to represent Forster resonance energy transfer. And then it's so excited that the high energy electron pops out. That's the job of light, to get these electrons in P680 to a high energy state. Now, this next part is a lot, but again, I encourage you to take the time to draw this out with your hand on a piece of paper. Why? Because it makes it stick in your brain. The more time and the more ways you allow your mind and body to process information, the better it's going to stick. And this is a lot. So here's our photosystem II that we just walked through. Again, here is our the purple is our hydrophobic thylakoid membrane. We have these 1, 2, 3 electron transport chain proteins. Then here's photosystem I and another coenzyme right here, another protein embedded in the thylakoid membrane. So, right, we just did photolysis and we have this high energy electron. What's going to happen next is this high potential energy, PE says potential energy, electron is going to move through this electron transport chain in a stepwise, losing potential energy as it moves from one to the next. So it's going to get accepted, right? The electron is going to get accepted, then it's going to be given away. And when it moves from an area of high potential energy to lower potential energy, some energy will be released and it's available to do work. What work? So, right, that's this right here. That available energy is going to be used to pump protons from the stroma up here. There's a proton pool up here to pump those protons through this center electron transport chain protein into the lumen of the thylakoid. So we are going against the gradient. That's why it takes energy. So we are building up an area of high proton concentration, a proton gradient here in the lumen. All right. Then once it's gone down, it's going to come over here to almost a PlayStation 1, Photosystem I, and get re-energized here. So, and it has a new name. The chlorophyll dimer in Photosystem I is called P700. Light, again, is going to hit these antenna pigments, just like our tennis player analogy. It's going to vibrate and resonate. We're going to do fret, right? Energy transfer through these pigments and pop that electron back up to a higher energy. Okay, so it's going to be re-energized by light in Photosystem I. Then Photosystem I is going to pass these electrons off to this coenzyme, which is going to, these high potential energy electrons are going to reduce NADP+. So NADP+ is the oxidized form. It does not have electrons. It is going to accept these electrons from Photosystem I that had just gotten re-energized. So now it has high energy electrons. It's going to get 2 electrons and a proton from up here in the proton pool to become NADPH. So, hooray, we have a reduced electron carrier. NADPH! All right. So, quick, let's do some NADPH accounting. Because for me and a lot of people, the math needs to math. And this is one part that high school level or even early college level textbooks, they either glaze over, leave out, or it's too confusing and nobody understands it. So let's just do some quick accounting. So if I start with 2 water molecules, right? So this is from our first board. We're going to get 4 electrons. This is in photolysis, right? 4 protons and oxygen. The electrons are going to go to the electron transport chain. The protons are going to build up in the lumen, and the O2 is going to diffuse out as waste. Now, let's look in our electron transport chain, right? If we take 2 of those electrons from the electron transport chain, 1 proton that's in the proton pool of the stroma, add it to NADP+, we're going to get NADPH. But that seems weird. Shouldn't it just be 1 electron would make it NADPH, right? Because it would make that plus 1 electron and H. But again, just like in the last thing, we need to do a quick zoom in on the physical structure of the nicotinamide ring in NADPH. So this is just one part of NADPH. NADPH is a much bigger molecule, but this is the part that does the electron accepting. So that's why we care about it. It is a ring structure with mostly carbons, but 1 point is a nitrogen. It's got 3 double bonds, and the nitrogen is positively charged. Why this is important is this is where the space is available to be adding stuff onto, right? So this is NADP+. This is its structure. We are going to with the help of that coenzyme, like on the last board, that is going to break this bond right here, and now making space available for one proton plus one electron. We'll add another H up here. So that's where one electron and one proton goes. Then down here, right, this is positively charged, we're going to add the other electron, and that's going to neutralize this positively charged nitrogen to be neutral. So that's how this structure physically carries those electrons and protons. Right? But what the heck is NADPH for anyway? Like, why do we care? This is my number one question, especially when it comes to biochemistry, because it just gets so... All right, so why do we care? We care because NADPH is a reduced electron carrier. Well, where is it carrying? And it's carrying 2 electrons and a proton to the Calvin cycle, which are also called the light independent reactions. It's the second part. It's the carbon fixation part of, like where we actually take the CO2, and the H's in glucose come from the NADPH. It comes from that, the protons and electrons that are being carried by NADPH. So check out this setup. I've got coupled reactions happening here. So we have NADPH. This is happening in the stroma, in the Calvin cycle. It is going to let go of some of its electrons. It's going to, right, oxidation is losing. So NADPH is going to oxidize to form NADP+. Those electrons are going to leave NADPH, and there is a 3-carbon molecule called 3-PGA, which is produced after the carbon fixation part of the Calvin cycle. It is going to, reduction is gain those to form those electrons that NADPH is giving away, and it is going to then transform into G3P. So G3P is reduced, or really 3-PGA is reduced to form G3P. Then G3P, eventually 2 of those are going to come together. There's going to be a lot of enzyme cut and paste in the stroma later, but that's going to form glucose C6H12O6. And right, because what's coming into the stroma is just CO2. So where do those H's come from? Those H's come from NADPH. Okay. Oh, it's all coming together. That's how glucose is actually made. Yes. Yes. But we're not done. We're getting close though. Don't worry about it. So remember, one of the main things the electron transport chain did was help us establish the proton gradient in the lumen. So again, here is my thylakoid membrane. Up here is the stroma, and down here is the thylakoid lumen. Now, you may be like, excuse me, your ATP synthase is upside down. Okay. ATP synthase is a 3D molecule, and it can go in any membrane it wants. But really, you gotta, one thing that's hard about working with diagrams is that we sometimes get really stuck on, especially when we've like learned the cellular respiration version of ATP synthase, it freaks us out to see ATP synthase go in a different way. But I mean, just like you can take a piece of paper, right, and you can read it a bunch of different, like, things in 3D space can move around, and that's okay. What's important is with our ATP synthase that we have the first portion of it, the entry, sort of the entry portion of it that the protons go into is facing the lumen. So you could turn this sideways because, right, the thylakoid is it's like an oval. It's like a 3D pancake with jelly inside, right? And the jelly is the lumen, right? And then the outside is the stroma. So, right, you could imagine this going all around the outside of the thylakoid. So, but I'm operating from that very first diagram. So in that case, the high proton concentration is the part where the entrance is, and then the exit is going to be towards the stroma. So that could be in any direction, but in this case, it's towards the stroma. That's the important part. So, protons get into the lumen two ways. One, through the electron transport chain, right, pumping them through, and two, through photolysis at the very beginning in Photosystem II, right? The electrons will move into the electron transport chain, but the protons stay in the lumen. So we're building up this high concentration, so we have a higher charge and we have a high potential energy section inside the lumen. Then the protons are going to move from the lumen through ATP synthase and into the stroma. All right? The energy is going to shift as these shift. So here we have our energy as potential energy. Just like when a dam holds back water, it's creating potential energy, right? Then as the protons flow through, it's going to physically turn ATP synthase. So we are going from potential energy to kinetic energy here in ATP synthase. ATP synthase is an enzyme, so it is going to take ADP and an inorganic phosphate. So ADP is 2 phosphates plus a third phosphate and make ATP, adenosine triphosphate, that has 3 phosphates here in the stroma. So that this ATP is chemical energy that we can use to do cell work. Hooray! But what is that work? In this case, this ATP is going to be used in the Calvin cycle to help us make glucose. So, in that process, right, this process of the movement of protons, the flow from an area of high concentration to low concentration through ATP synthase is called chemiosmosis. You see that also in cellular respiration. So what did light accomplish? We're wrapping it all up here, right? Light took our electrons that originally came from the photolysis of water, and with our friend Fret, not Fert, Fret, increase the potential energy of electrons. Then these high potential energy electrons went through the electron transport chain, forming the proton gradient. Then we reduced NADP plus to NADPH, and the proton gradient gave us the potential energy for chemiosmosis that drove the production of ATP. So and then let me just make sure, what do we say, right? Light raises the electron's potential energy. Yes, electron movement created that H plus gradient. Yes, we reduced NADP plus to NADPH. Yes, H plus gradient then powered ATP production. Yes. And then our NADPH and ATP are going to go to the Calvin cycle to help turn G3P, or to form G3P from 3-PGA. Yes. And then eventually that's going to make glucose, which in our simplified equation, hooray, is like our final goal product. So knowing that light is essential for photosynthesis, you got to know that. But that's the very base level. We're in AP biology here, or college biology. So you're at a can-do level when you can explain this whole thing or any diagram, and you can explain all the pieces, parts, and what they're doing and why. Why are they doing it? What's being achieved? but you're going to be fluent and really able to use this on the AP Biology exam to an AP Biology level when you can accurately predict what happens if something in any of those steps goes wrong. If something goes wrong, how is that going to impact the production of any of the intermediaries or the final products? So, and how do we get fluent? We practice. Practice, practice, practice. I hope this video was helpful. I know it was a deep dive and it was a lot, but that's why YouTube gives us pause, rewind. You can watch it a million times. You can fast forward through the parts that you totally understand. And so please do use this video as a tool to help you go from no to can do to fluent. If you found this video helpful, please give it a like. Subscribe to my channel for more chemistry foundations, quantitative skills in AP Biology. What else do I have? Tons of graphing, like teacher tips, all that kind of stuff. Leave a comment, a question, or an emoji down below. If you made it this far, you are a champion. Leave a sun, and I'll be sure to give a heart to your comment. And if you're a teacher or a student, check out my resources on Teachers Pay Teachers. My store is called Know and Grow with Gnomes. Again, I have tons of quantitative skills, so everything that is on the formula and equation sheet, graphing practice, like FRQ practice, just tons of different things to help you practice your skills and build your AP Biology fluency. Check it out there. Keep learning, keep growing, and we'll see you next time.